Hello students, welcome to the last session of chapter 1 of your biology NCERT, that is living world. So far in last classes what we have studied is about living things, their criterias, how to name them, how to scientifically name them, their classification, taxonomic categories and how hierarchically we can arrange them from species until kingdom. So as I have told in last class, I told you that we will be studying about 4 different organisms as well as their complete classification. So here on the board I have already written 4 different organisms. One is man, housefly, mango and wheat, where you can see I have written the complete classification of all the organisms. Now in the first column you can see that I have written common name, scientific name, genus, family, order, class and phylum or division. So we are starting here from the lower category. As you move downwards you are reaching the higher category. So we will start with man. Man, that is the common name, and the scientific name is Homo sapiens. That is Homo sapiens, where Homo is the genus name and sapiens is the species name. So the genus name is Homo. Human being belong to family called Hominidae. So this is Hominidae and order Primata, class Mammalia and phylum Chordata. So that is the particular way of classifying human beings. Once again to explain human being or man, scientific name is Homo sapiens. The genus name is Homo. This belongs to family Hominidae. The order is Primata, class is Mammalia and the phylum is Chordata. That is the classification for human beings. Next classification is one more organism, that is housefly. So housefly has a scientific name Musca domestica. Musca domestica is the scientific name of housefly, where Musca is the genus and domestica is the species. So Musca here is the genus name. This belongs to family Mucidae. Mucidae is the name of the family to which the housefly belongs. And Diptera is the order. Diptera is the order. Insecta is the class and Arthropoda is the phylum. So once again let me explain. Housefly, scientific name is Musca domestica. Genus name is Musca. They belong to family Mucidae. They have the order Diptera. They belong to class Insecta and to the phylum Arthropoda. So 2 of the organisms are animals, what you have studied the complete classification of man as well as housefly. Next is you will be studying about 2 different plants. Coming to the common name, that is mango. Mango scientific name is, we have learnt about this in our last classes, Mangifera indica. Mangifera indica is the scientific name of mango. So Mangifera is the genus name. This belongs to family Anacardiaceae. Anacardiaceae is the family name. Sapindales. Sapindales is the order. Anacardiaceae is the family, Sapindales is the order. And Dicotyledonnae. So Dicotyledonnae is the class to which the mango belongs. And last one here, remember that here it doesn't come phylum. Instead of phylum, as we are representing a plant, here you have to call it as division and it is Angiospermae. So mango can be classified as mango, the scientific name is Mangifera indica, where the genus is Mangifera. And when it comes to the family, it is Anacardiaceae and Sapindales is the order. And when it comes to the next category, that is what is class, that is dicotyledonate, and the last, that is division, is angiospermae. So that is the complete classification of mango. Next one is about wheat. So when it comes to wheat, the scientific name of wheat is Triticum aestivum. Triticum aestivum is the scientific name of wheat, where Triticum is the genus name. Poaceae is the family and the order is Poales. The class is monocotyledonate, and here the division is same as that of mango, that is angiospermae. Once again, to study the classification of wheat, wheat scientific name is Triticum aestivum. Triticum is the genus name, Poaceae is the family, Poales is the order, monocotyledonate is the class, and the division is angiospermae. So this 4 classifications are very much important based on your exam point of view. Why? Because one out of this is a compulsory question in your test as well as examinations. So here that is about 4 different organisms whose complete classification you have studied in today's class. One is man, housefly, mango and wheat. In all these, you might have noted while I have written the scientific name, I have underlined the genus and species name separately, as well as the genus first letter in each of the scientific name I have written capital, following the rules of nomenclature. So that's about 4 different organisms as well as their complete classification. Next topic is taxonomic aids. When it comes to taxonomic aids, here we will be discussing about different types of organisms which have been identified, classified as well as given a binomial nomenclature. Some of the biologists also have suggested that it can be used by, or it can be used for study purpose by preserving the organisms. Even the dead organisms can also be used for study as well as the living organisms if they are properly classified and proper nomenclature is provided. So there are different types of taxonomic aids which you are going to study. So first one out of that will be herbarium. So in herbarium what we do is, you select a small plant, maybe this small plant which is or which should contain flowers in that, and that part of the plant will be cut. Once that part of the plant will be cut, it will be placed within the newspaper. That is, if the newspaper opens up like this, the plant will be kept like this and it will be folded. Above this one, weight, that is big books or which is having more weight, that should be kept so that the plant which is placed between the newspaper spreads properly. The flowers, the petals as well as the leaves and the stem will be spreaded properly. And while you select a plant for this purpose, you should not select a thick woody stem. So the chances of drying these plants will be very much long compared to a herbaceous stem. Once it is dried properly, next what should be done is you have to keep on repeating, that is the newspaper should be changed every day once it is very soft stem, because it starts to excess or leave the water outside. That is why the newspaper should be changed. Or else what will happen is the plant will be affected by fungus. So it will destroy your whole herbarium process. After 1 week, you can change the paper for once in 2 days, because the release of water from these plants will start to reduce. So this process will be continued for almost 15. If it is summer, it will take very less time, that is maybe within 10 days only you can get your herbarium ready. But if it is winter or rainy season, it will take a little bit longer time because of the environmental condition. Once the plant is completely dried, next what you have to do is you have to stick it in a herbarium sheet. When it comes to a herbarium sheet, you will be sticking the plant like this. So imagine that the flowers are here and the leaves are present here. So the plant will be stuck to this herbarium sheet, and below this you can write the description. And at the right corner of the herbarium sheet, here the person or the date of collection, the time of collection, as well as the details, that is the family, the scientific name, the common name will be mentioned here. The reason why date will be written, or the collection time and date will be mentioned here, because to identify the season of the flowering, whether it is flowers throughout the year or only at a particular season, so that these sheets, once they are ready, they are kept under wooden boxes or kept inside wooden boxes, where they are arranged according to the classification, according to the proper classification, whether they belong to higher types of plants or lower kinds of plants. Based on that, it will be easily arranged. That is how you maintain or produce a herbarium, and you can use it for study purpose. Here you are using a dead plant for study purpose, which is preserved. Next taxonomic aid is botanical garden. In case of botanical garden, what will happen, or what we do is, we use or we grow here live plants. You are using live plants for study purpose. In this garden, more preference will be given to flora, that is plant. Both exotic as well as indigenous plants will be taken into consideration. So each plant will be having a nameplate, and when you see this nameplate contains the common name, that is the regional or the local name, what it is called in that particular area, the scientific name along with the family. So the board will be having 3 different lines. One will be with the common name, second will be with the scientific name, and third one will be the family the plant belongs to. So one of the biggest botanical garden, that will be Kew Botanical Garden, which is placed in England. In India, it will be Indian Botanical Garden, which is present in Howrah. One more Indian Botanical Garden which is present in Lucknow, that is National Botanical Research Institute. This is placed in Lucknow, that is India. So these are the 3 famous botanical gardens which are placed in India as well as in England. Kew Garden, which is placed in England, Indian Botanical Garden, Howrah, India, and National Botanical Research Institute in Lucknow. So that is about the second taxonomic aid, that is botanical garden. Next, third taxonomic aid, that is museum. When it comes to museum, here normally we'll be storing the dead organisms. We preserve them and store it so that we can use it for study purpose. Especially these museums are the one, normally any students are taken for educational too, so that they have an idea about the particular organism, whether it is living or dead. When it comes to museum, there are different ways of preserving the organisms. When it comes to a small organisms like a lizard or a rabbit or a bat, what we do is, there will be preservatives, that is a fluid in which this preserved organisms or the organisms can be stored. That preservative or the liquid in which the organisms are stored, that is called formalin. Inside formalin, the organisms will be stored. That is one way of preservation. Second way of preservation is called dry preservation, especially by pinning. If you see butterflies and all, if the formalin is added to the butterfly, they will start to decolor their wings. So the chances of the butterfly getting damaged is much more. That is why insects or butterflies which lose their color when we add the preservative, such type of organisms are dry stored. Normally we pin them in a wooden board which contains the thermocol. So we pin the organisms or insects inside the wooden board, that is the glass wooden board, so that you can easily observe the organisms. That is the second method of preservation. If you see a lion, tiger or a bear, neither you can you cannot use large amount of formalin to store them. Why? Because it is difficult to preserve them. That is why here also dry preservation is preferred. What we do, the organism, once they are dead, the internal organs will be removed and stuffing will be done. Once the stuffing is done, it will be sieved as the way the body of the organism was, and then it will be preserved as it is. That is the type of dry preservation. So you might wonder whether the skin might not be affected with fungi. Of course it will be affected. That is why formalins will be sprayed on the skin. Last type of preservation, that is skeletons. So once the organism is dead, the muscles, nerves, everything will be removed. The bones will be preserved as it is, and it will be arranged accordingly using strings. So all this are the different methods or different types of methods or different forms by which you can preserve the organism and simultaneously study the organism. That is the third taxonomical aid, museum. Next, fourth taxonomical aid, that will be zoo. So in case of zoos, Mysuru is very much famous for zoos. You might have seen Mysore Zoo, where you are not preserving, instead protecting the animals. And in zoos, it is very easy. You can identify the food pattern, habitat types, as well as behavioral changes in case of animals. So live animals which you might find only in the forest will be available in the zoos, so that you can identify their behavior, food habits, etc. And it is a very easy method, so that you can easily observe them as well as get the idea what type of environmental conditions are necessary for these organisms to survive. And here zoos, human beings, that is, only are allowed until a restricted area. You cannot go much more further. Why? Because they are placed inside the cages. Good care of this organisms or animals will be taken by feeding them properly, by proper vaccinations, etc. So this is another taxonomical aid where you can easily study about living organisms. Next, fifth form will be key. In key, you will be identifying the organisms, that is plants as well as animals, based on their similarities as well as dissimilarities. Key is a kind of book. So if the organism is in your hand, or if the picture is in your hand, you will be using the similarities and dissimilarities which are present in the organism. So the series of characters will be given. You have to identify whether, now for example, if a butterfly is in your hand, the presence of antenna will be there. So you have to go to the book if there is any presence of antenna. So next there will be a link regarding that particular question. So here you will be taking, or based on similarities and dissimilarities, you will be identifying the plant or a animal. Next character or next requirement, I told you that it is a book which contains 2 characters. So here, as a particular question, now for example, if this is a book, here question number 1, presence of antenna. Question or second sentence, absence of antenna. Now you have a butterfly, I told. So here it comes presence of antenna. So in the bracket only they will be given. If the antenna is present, then go to question number 4. So like that, there will be a lead, that is the next hint. Just like your treasure hunt, how you get a hint from once you keep on solving the mystery. In the same way, here also, once you identify one character, so there will be 2 contrasting characters. These 2 contrasting characters are called couplet. So once this couplet, out of this couplet, one is the right answer, you will get a lead to the next answer. By this form, you can identify whether this organism is a butterfly or a not. Okay, so it is applied to smaller organisms as well as larger organism. And key is different for different taxon. If you are thinking of identifying the family of the particular organism, you have a different key book. If you are thinking of identifying the genus, you have a separate book. So different taxon have different key books. That is the 5th type of taxonomic aid. Next one is Flora. So flora accounts, it is identification and distribution of particular types of plants in a particular area. So here it is restricted to one particular area where you will be identifying and studying the habits as well as habitats of particular plant species, that is flora. Next one is monograph. Here in this monograph, you will be studying about particular taxon, that is only one category. You will be studying about either a species, either a family, either a order. That means you will be studying or concentrating only on one type of taxon, that is what is called monograph. And the last type of taxonomic aid is manuals. Here you will be studying about, or you will be identifying the species or particular types of species present or restricted to one particular area. So here it will be generally any species. When it comes to flora, it will be only plants. That is the difference between a flora as well as a manual. So we have studied so far different types of taxonomic aids, based on which we will be studying either the living organisms or the preserved organisms for our own benefit. So we have studied about herbarium, botanical garden, museum, zoo, key, flora, monograph and manuals. So these are about different types of taxonomic aids. That ends the chapter Living World. I hope you have properly understood the chapter. Thank you.